Good morning. In this video, we are going to discuss transport across cell membranes. Of course, this is a topic we have discussed previously in different circumstances, and you will find different videos on the channel that talk about transport across cell membranes. But this time, we are going to focus more on the types of transporters across the cell membrane. First of all, I have to remind you that the cell membrane is a double phospholipid layer and that the interior lipid molecules are hydrophobic and therefore they do not allow hydrophilic large substances to pass. And therefore, when do we need a transporter across the cell membrane? When we have a large polar substance, as we explained before, or a charged substance that is large in size. If it is a polar and small molecule, it would pass easily like water molecules. So we need transporters across the cell membrane to transport the large hydrophilic or charged substances or to transport the ions. So we will have transporters for ions like sodium ion, potassium ion, that are called the ion channels, and we will have transporters for large molecules that are called carriers. But today we are going to categorize the transporters into three main categories according to how many substances they transport and in which direction. But before starting this explanation, I have to mention that the transporters are usually protein molecules that are embedded across the cell membrane, as you see. And since they stretch across the whole thickness of the cell membrane, then we call them integral proteins. And why specifically proteins are used as carriers and as channels? Because if we consider, for example, the ion channels that are supposed to be specific and to allow the passage of one type of ions like potassium or sodium, then the interior core of this hydrophilic channel, this is of course a cross-sectional area in the channel, and this interior core will be made of amino acids that are hydrophilic, which means that they are either polar or charged, and therefore they would be great for the passage of the charged particles, since hydrophilic attracts hydrophilic or hydrophilic loves hydrophilic. You might be asking yourself, if I have a potassium ion, for example, that is positively charged, wouldn't it be stuck to a positively charged part of an amino acid or radical or group? This is very true, but this is why we say that the ion channels are specific, which means that if I have a positive ion, then the internal core of the channel should be well prepared to ease its passage and not to hinder its passage. So the interior charge or the internal charge should be the opposite, should be allowing it in a way to pass across the channel easily. There are many details in this field, but we are not going to explain deeper in order to avoid having the video very lengthy and in order to focus on the main pieces of information that we want to deliver today. We categorize the transporters across the cell membrane into uniporters, symporters, and antiporter. A uniporter is a membrane protein that will transport a substance in one direction only, and it is only one substance. We have just mentioned that, for example, the ion channels are specific, which means that every channel can allow the transport or the passage of only one type of ions. We also have another group of transporters, which are the symporters, and the prefix sim, this should be an M, symporter, means at the same time. So we have two substances that are carried across the transporter at the same time, and as you see in the diagram, in the same direction. In the antiporter, we have two substances, A and B, that are transported at the same time, but in opposite directions. That's why here the prefix anti means in opposite directions. The diagram to the right is more clear because it shows you here that we have one type of ions, or one type of ions actually. They have the same color. In the same port, you have two different colors because they are two different substances but carried in the same direction. And here again, you have two different colors, which means two different substances but in opposite directions. So we read the text and explain it. What are then the uniporters? The uniporters are membrane proteins that move a single type of molecules across the membrane in one direction. Most frequently, those uniporters are passive transporters, which means that they carry the substance down concentration gradient from a place or an area where they have higher concentration to an area where they have lower concentration, and therefore we say that they transport materials down concentration gradient. When we have a channel like this, a protein channel that allows passage of an ion, for example, down concentration gradient, so we don't need energy, and thus it is a passive process. It is a passive process, but we cannot say that this is passive diffusion. This is called facilitated diffusion because there is facilitation from the part of the channel. The presence of the hydrophilic channel around this ion allowed it to pass safely across the cell membrane. But we still have, this is the majority of the uniports and the majority of the channels. But there are some uniports, a minority, that do the same job, that allow the passage of one type of a substance in one direction, but against concentration gradient, which means the arrow here would be reversed. For example, from the inside, where we have lower concentration, to the outside, where we have higher concentration. This is against concentration gradient. So if we reverse the arrow, then we would be transporting the substance against concentration gradient. And therefore, since it is against logic, because molecules or ions or particles should pass from higher to lower concentration, then it requires energy, and then it is a kind of active transport. And this is what happens, for example, for hydrogen ions. We have seen this along the membranes of mitochondria and chloroplasts, because we had electron transporters, which are actually protein transporters, stretching across the membrane of the mitochondrion or of the plastid, and that pump hydrogen ions against concentration gradient to create a proton gradient. And you can, of course, refer to these videos in order to remind yourself or to refresh your memory about this. So most of the time, the uniports are passive, and they carry out a process called facilitated diffusion, and the substance moves down concentration gradient, down its concentration gradient, which means from an area of higher concentration to an area of lower concentration. And since we are going against concentration gradient, then we don't require metabolic energy, or we don't require ATP. The uniporter protein will facilitate this natural movement. The natural movement is the movement down concentration gradient. I've given you the example of ion channels, which required a channel or a transporter because they were charged, but we also have some transporters for molecules, which we usually call carriers, like the glucose transporter system. The glucose transporter system is present in the membranes of the red blood cells, and it allows the passage of glucose from the blood to the inside of the red blood cell. This is a type of facilitated diffusion, and we call it the GLUT system. But sometimes, as I've just explained, the uniport is active. So we have primary active transporter, and primary means that it is an active transporter which acts on its own. It does not require a previous procedure or process to occur before it. So from the very beginning, and we will explain this in detail. You will understand what a primary active transport is when we explain what a secondary active transport is. So as I've just said, like the hydrogen pumps, which are, for example, like the carriers, the electron carriers, because they are not only carriers in the electron transport chain in mitochondria and chloroplast, but they are also considered hydrogen pumps. They are uniporters, but they are active uniporters. They are uniporters. Why? Because they move only one substance, but they are active uniporters because they use ATP. And why do they use ATP? Because they move the substance against concentration gradient. Here we can see the uniports, the antiports, and the symports in a very colorful and bright diagram. We have the uniport that carries for example, this is the GLUT transporter or the glucose transporter. It is a uniport and it transports glucose from the extracellular space, which lies between the blood and the cell membrane, or between the blood vessels actually, and the cell membrane, where it is present in higher concentrations to the inside of the cell or to the cytosol, where we have a lower concentration of glucose. We also have the antiport, and the best example for the antiporters are the sodium potassium pumps. These are protein pumps, integral proteins that stretch across the whole thickness of the cell membrane and that transport two ions at the same time. And actually, they are transporting them against concentration gradient. So potassium is being pumped using energy from ATP to the inside of the cell. And the purpose of all this is to keep the concentration of potassium very high intracellularly for optimum function of the cell. Because you know that the main cation intracellularly is potassium. But at the same time, we will find that this sodium potassium pump or transporter will pump the sodium ions outside against concentration gradient using energy from ATP. Why is it doing this? Because normally sodium should be the main cation extracellularly for proper functioning of the cell. But you might also have noticed that this sodium potassium pump or transporter pumps two potassium ions inward while or in exchange for three sodium ions outward. So here we have three sodium ions. Don't look here, this represents the concentration of sodium extracellularly, but here at the beginning, we have three sodium ions that are pumped extracellularly for two potassium ions that are pumped intracellularly. This achieves the goal of having sodium, the main cation extracellularly, and potassium, the main cation intracellularly, but the difference is meant because we need the outside, the net charge outside to be more positive than the inside to establish an electric gradient or a potential electric potential gradient across the cell membrane. And this is what we call the resting membrane potential. Of course, there are other ions, positive and negative outside, and other ions, negative and positive outside, but I'm talking about the net difference in charge. In case of the symports, like here, for example, we have a transporter that is called the sodium glucose porter. This symporter carries glucose against concentration gradient, because if you look at the direction of the arrow, first of all, why it is a symporter? Because it is carrying two substances at the same time in the same direction. One of the two substances, which is glucose, is carried against concentration gradient. How did I know this? Because in the outside, in the extracellular space, we have only a few glucose molecules, and in the cytosol, we have much more glucose molecules. So this requires energy. This should be an active process. How do we get this energy? We get this energy from the transport of sodium ions at the same time. But if you look here, you will find that the concentration of sodium outside is higher than the concentration inside. So sodium is being transported down concentration gradient, and it cannot, of course, diffuse outside the membrane outside the transporter by simple diffusion, because it carries a positive charge and it is charged. What's the relation between the passive transport of sodium, which is a facilitated transport in such a case, and the active pumping of glucose, and why should they occur at the same time? Because when these sodium ions are transported down concentration gradient across the transporter, then there will be energy released, and this energy that is released will be used to pump glucose against concentration gradient. But you might be wondering, how did the sodium ions release energy? This simply means that they had already stored energy from a previous process. And we can assume that the previous process was active pumping of sodium, because when you actively pump sodium, first, by the sodium potassium pump, you are using energy, and this energy is partly stored in the sodium ions as potential energy. So when they are passed down concentration gradient, they release this potential energy, which will be used to pump glucose. So we are going to talk first about the symporters. A symporter is a type of carrier protein that will transport two different molecules or ions, molecules and ions, across a cell membrane simultaneously in the same direction. And most of these transporters, symporters, represent secondary active transport. Why is it secondary active transport? Why? We all know now that the transport of glucose across the membrane is an active transport. But why is it a secondary active transport? Because the energy that it is utilizing for pumping against concentration gradient is provided by means of storage of energy through a primary active transport, which was the pumping of sodium. So we have a previous primary active transport that stored energy inside the sodium ions, and when the sodium ions were transported, they released this energy. So the secondary active transport, the energy means what? The energy to move one molecule against its concentration gradient, which is here the glucose molecule in our symporter. It comes from the movement of a second molecule down its own pre-established electrochemical gradient created by a primary active transport. So we can start here. We can reverse the text and start here. The primary active transport is the active pumping of sodium outside, and then there was an electrochemical gradient generated. There was a chemical gradient because the concentration of sodium outside here was higher, and there was an electric gradient because the positive charge outside were more than the positive charge inside. And this energy stored by this electrochemical gradient will be released in case of the symport. So this is the most common use of the symporter. So the sodium glucose symporter, first of all, is an example of secondary active transport. And second, you should know that this is the way we transport glucose in the kidney and or across the kidney cells, the tubules, and across the intestines. It is important to know this because we have just talked about another type of transport, which was just facilitated diffusion and which occurred across the membranes of the red blood cells. And you can thus deduce and reach an important conclusion that glucose is not transported the same way across all membranes or cell membranes in the human body. So this symporter we have just talked about is the sodium glucose linked transporter. We call it linked because the active transport of glucose is linked to the facilitated diffusion of sodium, and this is which we abbreviate with this acronym, which is SGLT1, because there is another sodium glucose linked transporter too. So actually now we have the GLUT transporter, which is by facilitated diffusion, and then we have two sodium glucose linked transporters. The SGLT1 will move glucose into the cell against concentration gradient, as we have just explained in this diagram. Glucose is pumped inside the cell against concentration gradient at the same time as the sodium ions. So they are linked, but the sodium ions are moving down their electrochemical gradient using the energy from the sodium's downhill flow. Downhill flow means from the movement of sodium down concentration gradient, or from higher concentration outside to lower concentration inside, while glucose is being pumped or pulled uphill into the cell. This is a kind of secondary active transport, as we have just explained, because it relies on previously existing ion gradients. And that ion gradient, in case of sodium, was set by an active pump like the sodium-potassium pump. And we call it a sodium-potassium ATPase pump because the sodium-potassium pump pumps sodium and potassium against concentration gradient in opposite directions. So because it's a pumper then, or a pump, then it needs energy. And that energy is achieved by breakdown of ATP, by hydrolysis of ATP. So the sodium-potassium pump should have a hydrolase activity, or like an enzymatic activity to catalyze the hydrolysis of ATP, rather than consuming cellular energy for glucose transport. So here we are making use of the energy of the sodium-potassium pump that has been stored in the sodium ions to pump the glucose. If we hadn't done this, then we would have utilized energy from the inside of the cell, and this would be waste of energy. So again, how does the SGLT1 and the similar symporters work? First of all, we would rather write a better title for this slide, which is the characteristics of the SGLT1 symporter and all the symporters. First of all, they are co-transporters because the symporter will bind. It has domains to bind with the sodium ion and the glucose molecule at the same time. And then the other characteristic is that there is a downhill gradient. The sodium is moving, and we have explained all this. This is just a recap of what we have said before. The sodium ion is moving from a higher concentration outside the cell to a lower concentration inside the cell, down its concentration gradient. And then the third characteristic, which is very important and which we have talked about in case of the mitochondria and the chloroplast, that we have a kind of energy coupling here. This means what? This means that we take the energy from one source and we pump it into another source. So we couple this release of energy from sodium to the pumping of energy into glucose to pass across the cell membrane against concentration gradient. So the energy released by the sodium ions moving down their concentration gradient will empower the SGLT1, the carrier, to simultaneously move glucose into the cell, even if glucose concentration inside the cell is already high. And this makes sense. Why does it make sense? Because we need to accumulate a good amount of glucose inside the cell, because this is our fastest and most abundant source of energy. And then we have a result. The result is the efficient absorption for glucose. And this is what I've just said. And this is a crucial process for nutrient uptake, because glucose is the most important nutrient in our cells, just like oxygen is the most important gas. So this is a crucial process in the gut, for example, when we want to accumulate glucose inside the cells of the gut for absorption later on. So we accumulate in which direction? We take the glucose from the lumen of the small intestine, from the cavity of the small intestine, transport it actively into the inside of the cells, and then to the outside for absorption by the blood. Similarly, as we've mentioned before, it's applicable in the kidney, and we've talked a lot about kidney reabsorption, and you can refer to the videos in the playlist for grade 10 biology, where you will find a lot about kidney reabsorption. You can refresh your mind, but of course we don't go into depth and we don't talk about transporters there. The SGLT1 is facilitating the movement, of course, of glucose, and it is considered a secondary active transport. Again and again, why is it considered a secondary active transport? Because it does not use ATP directly, but it rather uses the potential energy stored in the sodium ions because of the electrochemical gradient that has been generated. There is another co-transporter that is less famous, but that is very important, which is the potassium chloride co-transporter. These are transporters. They are membrane proteins, of course. They transport two substances at the same time. They transport potassium and chloride ions at the same time. You all know, of course, that potassium is a positive ion, whereas chloride is a negative ion. So they are co-transporters because they carry the two ions, they transport the two ions at the same time. But are they symporters or anti-porters? They are symporters because they transport the two in the same direction, like we see here in the symporter. In this symporter, they transport the two in the same direction. So they are electrically neutral because they do not generate an electric gradient. But so they are the potassium chloride co-transporters are symporters. But what is the role of potassium chloride transporters? Notice that they move potassium and chloride ions in the same direction. Here, for example, we can see the potassium chloride co-transporter. But we can also see that here, although to the left we have potassium and chloride being transported to the outside of the cell, to the right we have another type of transporter which transports both sodium and potassium to the inside as well as chloride. Then we have different types of transporters. This proves you that there are different types of transporters across the cell membrane, and that it depends upon the needs of the cell. Let's focus on this co-transporter. This co-transporter does what? It transports both potassium and chloride outside the cell. But remember that potassium has been pumped to the inside of the cell previously by the sodium-potassium pump, and that it is storing a lot of energy inside the cell. It is the main cation intracellularly. And this time, this transporter will allow it to release all the stored potential energy by moving down concentration gradient or downhill from the inside of the cell where it has a higher concentration to the outside of the cell. And meanwhile, while potassium is being transported down concentration gradient, just like we said about sodium in the sodium glucose transporter, it will release energy, and this energy will be used in order to pump chloride against concentration gradient. Why are we saying against concentration gradient? Because actually the concentration of chloride outside the cell extracellularly is higher. So you're moving chloride via this transporter from the inside of the cell where it has a lower concentration gradient to the outside where it has a higher concentration gradient. And it seems that sometimes this transporter has to function on purpose in order to prevent accumulation of chloride in particular inside the cell. So the purpose of the potassium chloride co-transporters is regulating the cell volume by preventing swelling, because we all know that if chloride accumulates inside the cell, it will attract water and there would be swelling of the cell. The second purpose is to maintain ionic balance, because this way you're keeping chloride outside high and chloride intracellularly low. So you're not just adjusting intracellular chloride levels at relatively lower levels than outside, and this way you are also doing another important function, which is preventing swelling. So once again, by pumping chloride outside using this co-transporter, you're preventing the accumulation of chloride inside, you're keeping the ionic balance, you are preventing swelling by absorption of water. This is very important, especially in nerve cells for neuronal function, and especially in GABAergic signaling. We will discuss this in the nervous system. There are receptors in the neurons that are called GABAergic receptors, and there are GABAergic neurons that release this substance, GABA, and we will discuss this in the nervous system, as I said. But in general, the potassium chloride co-transporter is important for all the cells to prevent swelling inside the cell, but in particular, this is very important for the neurons and the nerve cells. And there are four main isoforms. Isoforms are proteins that are a little bit different from each other, but that are close to each other. And we saw this in molecular biology when we were talking about the processing of messenger RNA, the alternative splicing, and how we can get different isoforms. You can refer to the process of alternative splicing of messenger RNA, sorry. If there are mutations in the potassium chloride co-transporter, are there mutations that are possible? Yes. because the co-transporter is a protein. It is encoded by a gene. And if this gene is mutated, then you will have a mutated transporter, and you might have problems in the concentration of salts inside the cell. And this is related to many hereditary neuropathies and diseases. So this process is a form of secondary active transport, as we have just explained, because in order to pump chloride extracellularly against concentration gradient, you need energy that is derived from the release of the potential energy that was stored inside the potassium ions after they have been pumped by the sodium potassium pump. So we will explain here step by step how it is an example of secondary active transport. We have maintained high intracellular potassium by the sodium potassium pump, and we said that this pumping of potassium and sodium by the sodium potassium pump is a primary active transport because it was not preceded by another type of active transport. And it was meant because we need potassium to be the highest ion, or to have a higher concentration intracellularly, while potassium, we want it to be the main cation extracellularly. But afterward, there will be a passive outward flow of potassium down chemical gradient, but this time along the co-transporter. And then at the same time, we will have simultaneous pumping of chloride ions out, and it is active because the chloride is already very high out, and then we are pumping it against its concentration gradient. Of course, we need energy. This energy is not taken directly from the cell, but it is done using the energy that was stored in the potassium gradient, and therefore we are ensuring a low concentration of intracellular chloride in mature neurons. Now we are going to focus on the differences between the GLUT1 transporter and the S-GLUT1 transporter. As I said before, there are several transporters in the body, so we don't only have a like a sodium glucose linked transporter 1, but we do have another sodium glucose linked transporter 2. But now we are focusing on the GLUT transporter, the glucose transporter, down concentration gradient passively, and the co-transporter. So now we are focusing on these two transporters only. So we have the sodium glucose linked transporter, like the type 1, that uses the sodium gradient to move glucose against concentration gradient, and therefore its function represents secondary active transport. I'm not explaining a new item, I'm just recapitulating what I've said on the diagram. Meanwhile, we have discussed the GLUT transporter that moves the glucose down concentration gradient. There is no active transport, there is no sodium involved, there is no previous primary active transport, and it is just by facilitated diffusion, which is a passive transport that does not require oxygen, that does not require energy. We have discussed the GLUT1 transporter that we have just explained, and I think we have seen it before. We have just explained it. Yes, we have just explained it when we were talking about the red blood cells, and we said that this is a passive transporter of glucose, and the difference between it and this one is very clear because here we need sodium ions. That's why we said on the slide that we do not need sodium ions. We have another type of transporter. GLUT1 transporter was used in the red blood corpuscles, whereas GLUT4 transporter is the glucose transporter that is dependent on insulin. So here we have a brief comparison. We have GLUT1 transporter that is used in the membrane of the red blood corpuscles and in the blood-brain barrier. We don't use sodium, of course, and it represents a type of facilitated diffusion. Whereas the sodium glucose link transporter 1 works in the intestines. We use sodium and it is a secondary type of active transports. And the question now is, why do we use all this? Why we have put GLUT4 apart, because GLUT4 is a separate entity that requires insulin. But now we are focusing on these two. Why? Because one of them is passive and the other is active. But why does the body need 2 or 3 or 4 types of transporters for glucose? First of all, they move glucose in different ways. So the sodium glucose linked transporter is a secondary active transport, and it brings glucose against concentration gradient, which means that it is capable of accumulating glucose. In case of the GLUT1 transporter that we saw in the red blood corpuscles, it was with or down concentration gradient, because we were just moving the glucose from the blood where it had a higher concentration to the inside of the red blood corpuscle where it had a smaller concentration. But when is it necessary to use the sodium glucose linked transporter 1? When we want to accumulate glucose inside the cell against the concentration gradient. So this will allow glucose to be absorbed efficiently. As I said before, we accumulate it into the cells from the lumen of the small intestine, or we accumulated it into the cells, to the inside of the cell from the renal filtrate. So this is very important to absorb glucose efficiently in the small intestine, or to reabsorb glucose efficiently from the kidneys. In the intestines, we use the type 1. In the kidneys, in the proximal tubules, we use the type 2. You can refer again to the videos on the playlist of grade 10, where you will find the anatomy of the kidney, the PCTs or proximal convoluted tubules, and how glucose is 100% absorbed actively from the proximal convoluted tubules, and the same occurs in the small intestine. But for the GLUTs, which act in the membranes of the red blood corpuscles, for example, and the blood-brain barrier, which is the membrane between the blood and the brain cells, glucose moves down its concentration gradient from the blood, where it has a higher concentration, to the inside of the cells. It is present in the red blood cells, in the brain, and these GLUT, as we have said, represent facilitated diffusion. We have GLUT1 in the brain and the RBCs. We have GLUT2 in the liver and the pancreas. And we have the one that depends on insulin, which is GLUT4. We call it insulin dependent, and it is present in the adipose connective tissue, where we need to convert glucose into fat, and in the muscles, where we need to accumulate glucose inside the muscles from the blood, because the muscles need this glucose for generation of energy. The second importance of the presence of two different transporters is that they allow both glucose absorption and glucose utilization. So they cooperate together to do both glucose absorption and glucose utilization. So in order to understand it, think of it as a two-step system. The active process, which is the sodium glucose linked transporter, pulls the glucose inside the cell. So it absorbs glucose from the gut into the blood, or it reabsorbs glucose from the kidney into the blood. So if there weren't these active, secondary active transporters, glucose would be lost in the stools in case of the gut, or it would be lost in urine in case of the kidneys. So from the small intestine, we return the glucose to the glucose we have obtained from digestion to the blood because we need it. And from the renal filtrate, after filtration of blood, we have filtered the toxic products, but unfortunately, glucose is also filtered, so we need to return it to the blood. So it's very important for absorption or reabsorption in case of the kidney. But afterward, the one that is after having safely returned the glucose to the blood from the intestines and from the renal filtrate, the passive transporter, the GLUT transporters, all of the types, will distribute the glucose to the cell. So after having accumulated glucose in the blood, the cells need to uptake this glucose for energy, and this is the role of the passive transporters, whether they are insulin dependent or insulin non-dependent. The third importance of having two different types of transporters is that they handle different physiological needs. And this is almost similar to number two, because the secondary active transport, SGLT, is for high efficiency absorption, because when you use energy, you are capable of uptaking the glucose from the lumen of the small intestine or from the renal filtrate and pump it into the blood. And this allows absorption of glucose even when the concentration of glucose in the small intestine or in the renal filtrate is very low, because the active process allows you to take these very small amounts of glucose from the gut or from the renal filtrate and return them to the blood, although the blood already has large amounts of glucose. So this is what we mean by high efficiency absorption, even when glucose is very low in the gut or in the renal filtrate. But the ones that are passive, they just regulate specificity of uptake by the cells. For example, about tissue-specific uptake, the brain needs constant glucose. So there are GLUT1 receptors in the brain. And since the brain cannot work without glucose, the receptors on the cell membranes of nerve cells should have a very high affinity to glucose. There should bind glucose strongly, even when the glucose levels in the blood are very low. In case of the liver, the liver needs glucose, of course, but the liver has another important function, which is to move glucose in and out depending on the fed and fasting state. If you are fasting, then the liver will convert glycogen into glucose and glucose will reach out of the cells. If you're fed, then the liver will take from the blood and store inside its liver cells. So it needs a transporter that has a low affinity, opposite to that of the brain. In the brain, we need a high affinity, like a magnet, to attract the glucose molecules, even if they are very low in the blood. The brain doesn't matter. It doesn't matter for the brain if there is a lot of glucose for the rest of the cells or not, because the brain is the most important organ. So it will take the glucose however low the quantities are in the blood. So it needs a receptor with high affinity. But when we have a receptor that is supposed to transport glucose in both directions and rapidly, so it has to be of low affinity. It doesn't have to attach itself strongly to the glucose, because this is against the process of rapid release of glucose. So in the brain, we have the high affinity receptors in the liver, we have the low affinity bidirectional receptors, and in the muscles, we have the insulin-dependent receptors because the muscles can take glucose only when insulin says so. So the only receptors that are in need of insulin are the receptors in the muscle. So this is what we call by tissue-specific uptake, which means that the different tissues have different strategies. And then we have the energy conservation. So you don't want every cell using energy to pull glucose in. So what do we mean by this? We mean that, for example, the enterocytes, which are the cells of the small intestine, and the kidney cells have those secondary active transporters because, as we said, they need to reabsorb glucose from the lumen to and return it to the blood, even if there are very small amounts of glucose in the lumen of the gut or in the lumen of the renal tubules. So we must consume energy because this is an important function to allow glucose to reach the blood, because the small intestine is the main source after digestion of glucose for the blood. But the other tissues do not need this active transport, and thus they do it passively. Why? Because the other tissues are receiving from the blood. So at any time, the concentration in the blood is higher than the concentration in the cells. The last importance is the regulation and the safety. If all the glucose transport depended only on passive transport, you couldn't be able to absorb enough glucose after a meal, because if you eat a lot of glucose during your meal, then the concentration in your gut might be higher than in the blood. And in such a case, you wouldn't be able to absorb the glucose if it were a passive process. And similarly in the urine, if there weren't this process of active transport, you would lose all the glucose in urine because the concentration in the filtrate or in urine would be higher than in the blood. And if you lose glucose in urine, and if you are not able to absorb glucose from the small intestine, then the brain and the red blood corpuscles might not get enough. So both together regulate the distribution of glucose between the tissues. So in one sentence, we can summarize that the sodium glucose transporters load glucose into the body. That's why they are present in the small intestine, where we have plenty of glucose inside the cavity of the small intestine after meals, and in the kidneys, because after filtration of blood, the filtrate does not only contain waste substances, but unfortunately, glucose can pass in the filtrate, and therefore we have to reload it into the blood once again. And after the glucose is loaded actively into the blood, the passive transporters will distribute it onto the body cells. The last thing we're talking about now is the examples of anti-porters. We have seen the anti-porters, and we have seen that the anti-porters are the ones that transport two substances at the same time in opposite directions. The sodium potassium anti-porter is easy to understand because it pumps protons outside the cell, but at the same time, sodium is being moved in. We will not go deep into the anti-porters, but you can understand if we pump potassium outside the cell, then we are trying to regulate the pH inside the cell. Because if potassium ions accumulate inside the cell, they will make the intracellular component more acidic. Therefore, the sodium-potassium antiporter is an antiporter that is very important to regulate the acidity. Here we can find the hydrogen ions that are being pumped outside to keep the pH inside, while the sodium ions go into the cell in exchange. Here you can see three different types of antiporters. You have the sodium-calcium exchanger, you have the sodium-hydrogen exchanger, and you have the chloride bicarbonate exchanger. All these three types are types of secondary active transporters. Here, for example, we can see that the calcium is being pumped extracellularly using energy that has been stored previously in sodium. Similarly, in the sodium-hydrogen exchanger, hydrogen is being pumped extracellularly using the energy that has been or that had been stored previously in sodium by the means of a sodium-potassium pump. The purpose in both conditions is to pump calcium outside the cell and to pump protons outside the cell. Pumping calcium outside the cell prevents unnecessary accumulation of calcium inside, which might affect cardiac function or muscle function in general and neuronal excitability. Pumping hydrogen outside is important in order to prevent accumulation of hydrogen ions inside the cell and therefore to prevent excessive acidity inside the cell. There is an important correction here that I have mentioned, or that I have to mention, is that although they are put on the same diagram, I don't know why they were on the same diagram, the chloride bicarbonate exchanger is not a type of secondary active transport. So I apologize if I've said this at the beginning because I saw them all on the same diagram. But the chloride bicarbonate is called the chloride bicarbonate exchanger is called a chloride bicarbonate exchanger, and it is a passive transporter, meaning that chloride is being transported down its concentration gradient because chloride has a higher concentration extracellularly, while bicarbonate is being shifted outward again down concentration gradient. So it's electrically neutral because you exchange one negative anion for another negative anion. And now I realize that they are put on the same diagram because they are all antiporters. So I apologize again. So it is a passive one. And the aim is to make the bicarbonate leave the cell in order to adjust once again the pH of the cell and to prevent excessive alkalinity of the cell. So we have mentioned the sodium hydrogen transporter, and this is very important for acid resistance. In some types of bacteria, they use this sodium proton antiporter to pump the protons, to expel the protons and pump them out in exchange for the uptake of amino acids like arginine. Why are they doing this? First of all, they expel the protons because they are present already in an acidic environment like the stomach, and they do not want the protons to accumulate inside and to make their cytosol very acidic. And meanwhile, they benefit from the exchange because they get an amino acid. Sometimes when the antiporter pumps calcium out, like we've seen in this antiporter transporter, it is important to maintain low calcium intracellularly, and this is very important for adjustment of cell signaling. In nerve endings, in the synaptic knobs, you can refer to human biology and to the nervous system. At the nerve endings, at the termination of nerve endings, we have swellings called the synaptic knobs that contain vesicles, and these vesicles contain the neurohormones called the neurotransmitters that are necessary for transmission of the nerve impulse. And these sodium proton anti-porters, or what we call the neurotransmitter transporters, are essential for accumulation of the neurotransmitters inside the vesicles in exchange of other ions that are being pumped outside. For example, in this diagram to the left, we can see here the synaptic knob, which is the swelling present at the end of a nerve or an axon. And this is one neuron, or the end of one neuron. This is the end of another neuron. And in between, we have a space that is called the synaptic cleft, as you know. And this place where the synaptic knob of one nerve ending ends or comes very close to the synaptic knob of another nerve ending, we call it the synapse. So we have a synaptic knob or two synaptic knobs. We have a synaptic cleft, and this whole area is called the synapse. You can see the synaptic vesicles that are surrounded by small membranes and that are full of neurotransmitters. Sometimes we want to reuptake the neurotransmitter and pump it again into the vesicles. You can see here what we call the neurotransmitter transporter that allows reuptake of the neurotransmitters and their accumulation in the synaptic vesicles, and in exchange there will be pumping of or release of sodium ions. So again, these transporters clear the synapse using the sodium gradient. So these anti-porters use the sodium gradient actually to, that's to say, the energy that is released from the passive transport of sodium to pump the neurotransmitter inside the vesicle. I know it's a bit complicated, but it's understandable. I hope you found this video useful. And if you found it useful, then share it with your colleagues and subscribe to the channel if you haven't already, and send your comments, and see you on the next video.